Hi everyone, I am Dr. Sheela, Assistant Professor, PG and Research Department of Microbiology, St.Joseph Arts and Science College, Autonomous, Cuddalore. Today I discuss about the topic plant growth promoting rhizobacteria, PGPR. What is PGPR? Plant growth promoting rhizobacteria occupy the rhizosphere region of many plants and have beneficial effect on the plant. Example of plant growth promoting rhizobacteria: Pseudomonas, Enterobacter, Arthrobacter. The beneficial effect may be direct mechanism and indirect mechanism. What is the direct mechanism? It is used to supply the nutrients, producing hormones, or indirect mechanism, stress resistant protection against disease manner. The rhizobacteria concept was first time given by Hiltner in 1904 to detect the zone of soil that surround the roots where the microbial populations are accelerated by root activities. The term of PGPR was proposed by Kiloeper et al. In 1980 and has been used for the long time, especially for fluorescent Pseudomonas involved in bacterial pathogen and biocontrol activity and enhance the plant growth promoting rhizobacteria. This one is PGPR activity, basis of location, extracellular, intracellular, basis of function, plant growth promoting rhizobacteria, biocontrol activity, stress heme regulating bacteria, basis of activity, biofertilizer, phytostimulator, rhizoremediator, biopesticides, bioprotectants. What are the types of PGPR? Extracellular PGPR and intracellular PGPR. What is extracellular PGPR? May exist in the rhizosphere and in the rhizoplane or in the spaces of the cell of the root cortex. The important bacterial genera of extracellular PGPR organism: Agrobacterium, Atrobacter, Bacillus, Calypbacter, Erwinia, Micrococcus, Pseudomonas and Serratia. Next one is intracellular PGPR. They locate inside the specialized nodular structure of root cell. They belong to the family Rhizobacteriaceae and includes intracellular PGPR organism Alorhizobium, Bradyrhizobium, Mesorhizobium and Frankia. Next one is mechanism of PGPR, direct mechanism and indirect mechanism. Direct mechanism, it is a biofertilizer activity, and indirect mechanism, the growth promotion and biocontrol activity. First one is direct mechanism. It facilitates nutrient uptake and nutrient availability. The direct mechanism includes nitrogen fixation, phosphate solubilization, potassium solubilization, siderophore production and phytohormones production. Nitrogen fixation, the PGPR provides nitrogen to plant by two mechanisms: symbiotic nitrogen fixation and non-symbiotic nitrogen fixation. Symbiotic nitrogen fixation, this is a mutual relationship between microbe and plants such as rhizobacteria and frankia. Non-symbiotic nitrogen fixation is carried out by free living associative and endophytic such as cyanobacteria, acetobacter. Next one is phosphate solubilization. Phosphate solubilization is another key element in the nutrient of plants. Organism bind with the phosphate solubilizing activity and termed as phosphate solubilizing microorganism and provide the available form of phosphorus to plants and thus constitute to chemical phosphate fertilizer. And then potassium solubilization. PGPR are able to solubilize the potassium through the production and secretion of organic acid. The example of potassium solubilizing organism Acetobacillus, Burkholderia and Pseudomonas. Next one is siderophore production. They are microbial chelating compounds. Hillhoefer in 1980 the first time described the importance of siderophore production and PGPR in the enhancement of plant growth. The last one is phytohormone production. Phytohormones such as auxin, gibberellins, cytokines effect on the cell proliferation. The IAA produced by plant growth promoting rhizobacteria, it has a positive effect on root growth and help in the cell division and differentiation. Next one is indirect mechanism. Indirect mechanism that is antibiotic production, hydrolytic enzyme production, induced systemic resistant exopolysaccharide production. First one is antibiotic production. The plant growth promoting rhizobacteria produce antibiotics against phytopathogens. It is a biocontrol mechanism. A variety of antibiotics have been identified including compounds such as benzene, tropholine and polymyxin, etc. Next, lytic enzymes. Plant growth promoting rhizobacteria produced some enzymes such as chitinase, dehydrogenase, beta-glucanase, lipase, etc. They exhibit hypersensitivity activity attacking pathogen by secreting cell wall hydrolysis. It protects from biotic and abiotic stresses. Third one is induced systemic resistant ISR. It is defined as a physiological state of enhancing defensive capacity in response to specific environmental stimuli, and as a result plant innate defense are activated. Many individual bacterial compounds induced systemic resistance such as lipopolysaccharide, siderophore and volatile compounds, etc. Next one is exopolysaccharide production, EPS. EPS production is the most important in biofilm formation and colonization. It helps to hold free phosphorus and circulating essential nutrients. The functions performed by exopolysaccharide producing microbes is a shielding from desiccation. Next one is a role of PGPR. Conversion of unavailable form of phosphorus into available form to the plants by secreting organic acid. Plant growth promoting bacteria also produced siderophore. This increased the iron availability. And then phytohormones such as auxin, cytokines, gibberellins are produced by plant growth promoting rhizobacteria which are induced the plant growth. The plant growth promoting rhizobacteria produce certain enzymes such as chitinase, lipase, protease, etc. Next, plant growth promoting bacteria can also able to fix atmospheric nitrogen and make it available to the plants. It controls the plant pathogenic bacteria by competing for nutrients, production antibiotics and antifungal metabolics. The induced plant mechanism against pathogen. When the entry of pathogen, this is called as induced systemic resistance, that is ISR, which protect the plant against pathogen by signaling. The abiotic stress are due to heavy metals in soil, the nutrient deficiency and salinity. PGPR microbes act as stress relievers. It is the stress relievers. The indigenous and native microbes are effective of competitive. Some of the plant growth promoting bacteria, example Pseudomonas, as used as a biofertilizer for many crops. Okay, thank you.